We know that lipids are part of our diet and are used to produce energy. So lipids have to be stored in prevision of time when energy is needed. Any imbalance between lipid storage and lipid usage will lead to disease. So for example, an excess of lipid storage due to a high caloric diet will lead to obesity or type 2 diabetes. In the previous video, we have seen that triacylglycerol is the main source of energy storage. They accumulate in specialized cells called adipocytes, and they are stored into these very large lipid droplets. So the goal of this video is to try to determine how is fat stored in and released from adipocytes. There is a first challenge. Triacylglycerol, you remember, are extremely hydrophobic, and they are accumulated in an hydrophilic environment, the cytoplasm. So how this challenge is overcome? Well, the solution comes from the architecture of lipid droplets. Lipid droplets are surrounded by a monolayer of phospholipids. And if you remember the structure of phospholipids, they have hydrophobic tail and an hydrophilic head. So the hydrophobic tails of the phospholipids are facing the triacylglycerol that is stored in the lipid droplets, while the hydrophilic heads are facing the outside of the lipid droplets, are facing the aqueous environment of the cytoplasm. So now we have a stable interface between the lipid droplets and the surrounding cytoplasm. Furthermore, some proteins are associated with a lipid droplet. In particular, a protein called perilipin form a layer that at the surface, at the outer surface of the lipid droplet. How are triacylglycerol molecules mobilized? Remember that triacylglycerol are formed of glycerol and 3 fatty acids. And these covalent bonds between glycerol and fatty acids have to be broken. This process is called lipolysis, and it's in fact a 3-step process. Each step is catalyzed by a different reaction. So the first step is the conversion of triacylglycerol into deacylglycerol. And this step is catalyzed by the adipose triglyceride lipase, or ATGL. The second step is the conversion of deacylglycerol into monoacylglycerol plus 1 molecule of fatty acid. And this step is catalyzed by an enzyme called HSL, or hormone-sensitive lipase. Finally, monoacylglycerol is broken down into 1 molecule of glycerol and 1 molecule of fatty acid, and this reaction is catalyzed by an enzyme called the monoacylglyceride lipase, or MGL. Now we have a second challenge. How is the timing of the triacylglyceride degradation coordinated with the energetic needs? These enzymes that are responsible for lipolysis have to be active for a very specific window of time when energy is needed. So we have 2 questions to answer. First, how this enzyme can be activated. Second, what determines the time at which this enzyme will be activated. So let's talk first about the activation of these enzymes, starting with an unstimulated adipocyte. The 3 enzymes are inactive because they are maintained away from their substrate. They cannot be recruited at the surface of the lipid droplet. For example, ATGL needs another protein called ABHD5 to be recruited at the surface of the lipid droplet. This protein is in fact maintained in an inactive form, bound at the surface of the lipid droplet, bound to perilipin. Similarly, HSL, which is bound to FABP4, another protein, is free in the cytoplasm and cannot be recruited at the surface of the lipid droplet. So what you have are two enzymes that are needed for the first two steps of lipolysis that are sequestered away from their substrate. And as a consequence, lipolysis cannot occur. Now, what determines the activation of adipocytes? We can take an example. An individual is engaged into an intense and sustained physical activity. The hydrolysis of triacylglycerol will be needed to provide energy to sustain this physical activity. The individual will produce hormones that will act on adipocytes and activate adipocyte. The consequence will be the activation of the enzyme involved in lipolysis. For example, hormones from the catecholamine family, such as adrenaline and noradrenaline, will control the activation of adipocytes. So now let's imagine that catecholamines are produced. What will happen? How adipocyte can be stimulated? In the membrane of the adipocyte, there is a protein, a receptor, called the beta-adrenergic receptor. This receptor has an extracellular domain that can bind catecholamine, and has an intracellular domain that can bind a trimeric G protein. When catecholamine is bound to its receptor, there is a change of conformation of the receptor that is transduced across the membrane and results in the activation of the trimeric G protein. This trimeric G protein will in turn activate another membrane protein, an enzyme called adenylyl cyclase, which catalyzes the conversion of ATP into cyclic AMP. So the consequence of catecholamine binding is an increase of the concentration of cyclic AMP. Cyclic AMP will then activate another protein, pKa, which is the protein kinase A. As a kinase, this pKa will then phosphorylate a certain number of proteins, including perilipin and HSL. So what is the consequence of these phosphorylation events in activated adipocytes? Let's start first with the phosphorylation of perilipin. When perilipin is phosphorylated, it will trigger the dissociation of ABHD5, which is now active and can recruit ATGL at the surface of the lipid droplet. The phosphorylation of perilipin will also create a remodeling of the perilipin layer at the surface of the lipid droplet and will increase the access of enzymes to the surface of the lipid droplet. Now, what about the phosphorylation of HSL? When HSL is phosphorylated, it becomes activated, and in complex with the protein FABP4, it will be recruited at the surface of the lipid droplet. So now we brought the first two enzymes in the lipolysis process in contact with their substrate. ATGL will be active, HSL will be active, diacylglycerol will be produced, monoacylglycerol will be produced, and now MGL, which is abundant and active in the cytoplasm, can in fact catalyze the conversion of monoacylglycerol into fatty acid and glycerol. The next question is, what is the fate of fatty acid and glycerol that were released by lipolysis? Free fatty acid will be bound to FABP4, which acts as a chaperone, helping the fatty acid to travel through the cytoplasm. Then the fatty acid will be exported into the bloodstream, where in association with serum albumin, it will travel to organs that need energy, for example, in muscle. The glycerol will diffuse in the cytoplasm and then will be exported to the bloodstream through a channel protein called aquaporin. And then in the bloodstream, it will travel to the liver where it will enter the glycolytic pathway.
Is there a chaperone for glycerol?
No, there is no need of a chaperone for glycerol because glycerol is hydrophilic and soluble both in the cytoplasm and in the bloodstream. That is in contrast with the fatty acid, which is in fact toxic for the cell and very hydrophobic. And so you need, for this fatty acid, a chaperone that basically not only protects the fatty acid, but also protects the cell. So in conclusion, we have seen in this video two new modes of enzyme regulation. The first one is a regulation of the activity by sequestering enzyme away from their substrate. And the second is by having hormones that will act on cells, will activate signaling pathway inside the cell, and this activation will lead to activation of enzyme.